I'm here to talk to you about DNA, deoxyribonucleic acid. As we mentioned in class, that is our genetic code. That is where we store information for how to build all the various things we need to make in order to survive. That's where every living organism stores the information they need in order to survive. One of the things that you doubtless know about DNA is that it's actually a polymer. Protein is a polymer, DNA is a polymer, RNA is a polymer. A polymer basically is when you take individual bits and pieces and assemble them into a long strand. You take the monomers, and in proteins, as you studied this summer, that monomer is an amino acid. In DNA, the monomer is known as a nucleotide. And nucleotides have three parts: a phosphate, a sugar, and a nitrogenous base. You assemble a series of these guys into a long strand, and then you assemble a second strand. Because DNA is not single-stranded, it's actually double-stranded. And you take those two strands and arrange them so that they run in opposite directions. But DNA, the way those polymers are built, it actually does have a direction. We have one end we call the 5' end. That's the end with a phosphate hanging off. And then we have one end we call the 3' end. That's the end that doesn't have an extra phosphate hanging out there. And so you have one strand with a 5' end going here and a 3' end at the other side. The second strand arranges itself so that they do not intersect, but they run one next to the other. Hanging off of these two strands are the nitrogenous bases that point towards the middle. And so you have basically two strands side by side with a bunch of stuff hanging into the middle. This whole ladder-like structure is twisted. And that twist we call a helix. Because there are two strands involved, we actually call this a double helix. You can see here three different nucleotides. We've got a thymine, which is represented by the letter T, an adenine represented by the letter A, and a cytosine represented by the letter C. These structures do have some similarities, right? They each have this little projection, which is actually a phosphate. They also have this sort of pentagon shape, which is the sugar, which is deoxyribose. And then they also have a nitrogenous base hanging off one side. For a polymer to form, what we need is not merely a small sort of random attraction between molecules. What we need is a genuine covalent bond, genuine electron sharing to happen between these guys. And you're not going to bond to just anywhere on a nucleotide. The bonds that form are actually going to form between the phosphates and the sugars, like so. When this happens, we create the zigzag structure, which you can see here: sugar, phosphate, sugar, phosphate, sugar, phosphate. That zigzag structure is what we call the sugar-phosphate backbone. Hanging off this backbone are all our nitrogenous bases. These nitrogenous bases for DNA can be one of four things: A, T, C, or G. Adenine, thymine, cytosine, or guanine. Now, they do have different structures, right? And those different structures are significant. The reason that those different structures are significant is because of a process we call base pairing. These basically act as puzzle pieces, so that when you get this other strand of DNA, it's got to match up perfectly with this strand. All right, so what just happened was that I built the anti-parallel strand. Well, at least I chose the nucleotides that would belong in it. And it's pretty easy to choose the nucleotides because of this phenomenon known as base pairing. Because these nitrogenous bases have unique shapes, they'll only match up with one other nitrogenous base. Which means that if you know what nucleotide is on one strand, you know the opposite nucleotide. C's and G's always match up, cytosines and guanines. A's and T's, adenines and thymines. Here you can see a T and an A, thymine, adenine. Anytime there's a T, you put an A. Anytime there's an A, you put a T. Anytime there's a C, you put a G. Anytime there's a G, you put a C. What happens in the middle here is actually hydrogen bonding. Because they are nitrogenous bases, that means that they've got nitrogen. And as you guys know from our discussion of hydrogen bonding, nitrogen is able to hold on to electrons really well. So we end up with a very electron-rich area and a very electron-poor area, so that you actually have a partial positive attracted to a partial negative in a very intense way, which is known as hydrogen bonding. C's and G's can actually form 3 hydrogen bonds. A's and T's can only form 2. So it's easier to break apart an A and a T than it is to break apart a C and a G. That said, there is no covalent bonding happening between these two sides. No covalent bonding. All right? That means that if you heat this up, or if you throw something in that can disrupt hydrogen bonds, you can pull apart the 2 complementary strands. All right, so we've got our base pairing happening. I set up these nucleotides at the other side. Let's take a look at these nucleotides. Phosphate, sugar, nitrogenous base. Phosphate, sugar, nitrogenous base. Phosphate, sugar, nitrogenous base. That is not anything different from what we had on this side. The difference lies in the directionality. The phosphates are pointed down this way, which means that our 5' end is opposite from what it was on the other side. Our 3' end, also opposite. Sort of makes sense. It is anti-parallel. Now the phosphate, which you see here, is going to have to connect covalently to the next sugar. To create our sugar-phosphate backbone, we still need covalent bonding. The sugar-phosphate backbone is where we are making our polymer. And so let's make those bonds. All right, that's it. That's the structure of DNA. We've got 2 anti-parallel strands that have been arranged according to complementary base pairing. The fact that they're anti-parallel was actually first discovered by Watson and Crick, the guys who discovered the structure of DNA. And they worked it out using like sort of a cardboard and wire model. But what I have to show you right now is actually a 3D animated model, so we can see that it's not just flat, it's actually a double helix. Double helix you see here is actually the 2 complementary strands twirling one about the other. Facing towards the center, you see step-like projections. That is where the nitrogenous bases meet. This twist you see is called a helix. When we studied protein structure, you found out about the alpha helix, and that was a twist of one substance. Here, 2 different strands are twisting. That's why we call it a double helix. Now, when we're talking about DNA as the code for life, basically what it does is it takes those A, T, C's, and G's and arranges them in such a way that they can communicate information. Just like a computer uses 1s and 0s, it uses binary to communicate information. Let's talk about the different pieces of DNA. We have some sequences called genes that encode proteins. We have some sequences called promoters that basically promote the expression of that protein. They say, here is where the protein is, you need to read it. The most famous one for us vertebrates, anyways, is the Tata box. Whenever your cell sees that, it knows that right afterwards is a gene. Right afterwards is the information you need to make a protein. And so that will be used in order to attract the machinery needed to read the gene. And when I say read the gene, I mean literally read the gene. Because you read each nucleotide. You say, well, there's an adenine, I better copy. That copying process we call transcription. It's how you go from DNA to RNA. Once you have your RNA transcript, you then read that RNA transcript and translate it into another language. By another language, I mean something that doesn't use nucleotides. Both DNA and RNA use nucleotides. They have phosphate, sugar, nitrogenous base, both of them. They use different sugars, and they have 1 different nitrogenous base, but they still only have the 4 different options. And they still assemble them on a sugar-phosphate backbone. But when you translate to a new language, and this language is the language of proteins, you're going to end up with a lot more options than just 4. And so you're going to have to completely translate it from that small little language of the nucleic acids into this wider one of the proteins. That process by which information goes, DNA, RNA, protein, that is the central dogma of molecular biology. It is basically one of the sort of founding principles of molecular biology. Let's talk about the third piece of the segment of DNA that we are going to be building in our modeling lab. We already talked about genes, we talked about promoters. The last one we have left are called telomeres. Telomeres are the protective caps at the end of your DNA, right? Our DNA is linear, meaning that it's a strand. At either end of that strand, we're going to have this protective segment called a telomere. The telomere is just a simple repeated sequence that provides like a little buffer so that your genes and so that your promoters aren't exposed to the wild world of the rest of the nucleus. By having that protective buffer at the end, if something were to happen, like say the DNA were to fuse to something it wasn't supposed to, it's not a gene that's impacted, it's just the end of the chromosome, which can still cause problems, admittedly, but it gives you a second layer of protection. Another thing that telomeres do is that they provide a little bit of extra sort of nonsense happening at the end of your DNA. This is significant because every time you make a new cell, you have to copy your DNA from a previous cell. When you copy your DNA, you can lose like 25 to 200 nucleotides every single time. And so when you lose those nucleotides, if they were significant, if they were encoding a protein, let's say they're encoding hemoglobin, now you got no hemoglobin in your new cell. That's a problem. What if they were encoding actin, which is important for structural reasons? Now you got problems. So in order to protect those kinds of genes, we've got these telomeres. Because if you lose 20 or even 200 random nucleotides off the ends, it's just a sequence there as a protective cap. So you're not losing information, you're just having a little bit less of that protective layer. As your cells get older, a normal part of the aging process is that your telomeres get shorter, because each time your cells replicate, you lose some of them. And so when you lose some of them, you're going to end up getting shorter and shorter and shorter telomeres. Once your telomere reaches a certain point, it tells your cell, you can't divide anymore. You're done. Cell division is done for you, which happens when cells get older. And if you're interested in the aging process, you might want to reverse that. And people who are interested in having cells that didn't age as quick and didn't die tried to find a way to keep that telomere long. And one of those ways is a protein that goes in and adds extra copies of that telomere sequence, of that protective little buffer. And so what they did was they activated this telomerase that added extra copies at the end of the telomere sequence so that the telomeres would always be long. In mice where they did this, there was a high incidence of cancer, which brings us to the idea of cancer. Cancer, you've got this uncontrollable cell division. In a lot of cancers, we have telomerase turned on when it shouldn't be. We believe the reason for this is because when you have a telomere, and it gets shorter and shorter over time, when it's too short, your cells can't divide. So for a cancer cell, which needs to divide like crazy if it's going to be cancerous, you're going to have to keep making those telomeres longer and longer. And so in cancers and in tumors where that telomerase has been turned on, you're going to get this uncontrollable cell division that's not held in check by the shortening telomere. All right, so we covered genes, Tau boxes or promoters, and telomeres as pieces of our DNA, as pieces, as sequences you can find on the DNA. They are not the only sequences. When we get into our genetics unit, we'll get into a lot more of those sequences. And how you copy over DNA and how you preserve DNA, these are ideas that we will definitely cover in class. For the modeling lab, it is important to understand how nucleotides are connected, why nucleotides are important, what kinds of things DNA can encode, base pairing, and how the structure of DNA is held together.